Thank you, Aldous, for the introduction. Let me just share my screen. I hope that you can see it, right?
Yeah, it's okay.
Okay, great.
Yeah.
It's a pleasure for me to be able to present the work of my lab to the fragile nucleosome community. Let me just move one of the windows from this screen to this one. Okay, I think this is now better. Okay, so what I decided to talk about today is a recent work from my lab about figuring out the landscape of in vivo methyl-cytosine recognition by transcription factors. But before that, I thought I could just give a very brief background about my lab and the kind of research that we do. So my lab, as Aldous mentioned, is located at McGill University in the beautiful city of Montreal in Canada. And we work on mechanisms of gene regulation. So there are two main pillars to the research that we do. One is on transcriptional regulation, and the other is on post-transcriptional regulation. So in terms of transcriptional regulation, we are interested in the functions of transcription factors, how they bind to the DNA, how they regulate the expression of their genes, how they interact with other factors that are involved in gene transcription. Post-transcriptional regulation, we are interested in the functions of RNA binding proteins, for example, their role in regulation of RNA splicing and RNA stability. And eventually what we do is that we try to apply the things that we have learned by looking at the basic mechanisms of gene regulation at the transcription and post-transcriptional level. We try to apply them to the context of human diseases, particularly cancer and neurodegenerative disorders, to understand what are the regulatory mechanisms behind these human diseases. And today, as I mentioned, I will be mostly talking about this pillar of our work, transcriptional regulation. And what we do here is basically try to understand how transcription factors work together with histone modifications, with DNA methylation, to recognize the DNA, bind to the DNA, and eventually to regulate gene expression. As you know, these relationships are super complicated. So transcription factors can affect histone modifications. Histone modifications in turn can affect the binding and recruitment of transcription factors. Transcription factors can affect DNA methylation by recruiting different factors to the DNA, et cetera, et cetera. So this is a very complicated and multi-way relationship, basically. And what I am focusing today is on this particular aspect, how methylation of the DNA is affecting transcription factors. So I will start by just briefly mentioning what is out there and what we know about recognition of methylated DNA by transcription factors. And when I say methylated DNA, I'm basically talking about methylation of cytosine in CpG dinucleotides, right? So this is by far the most abundant type of DNA methylation that we see inside the cells. And there has been this traditional view for quite some time that methylation of CpG dinucleotides is generally inhibiting the binding of transcription factors. So this can be either a direct effect, where the transcription factor that can bind to the DNA, when the DNA is methylated, it just simply doesn't recognize the DNA. It's not able to interact with that kind of methylated DNA. Or this can be an indirect effect. For example, when the transcription factor binding site is methylated, other proteins, such as methyl binding domain containing proteins, they bind to the DNA, and by doing so, they outcompete the transcription factor and basically prevent the transcription factor from binding to that DNA. But whereas this is the main basically mechanism that we've known for quite some time, and a lot of the transcription factors that people have studied, they seem to behave like this. There are obviously exceptions. So there are transcription factors that are known to actually bind to methylated DNA preferentially. This transcription factor CEBP beta is a prime example. I think so back in 2013, using techniques such as protein binding microarrays, people showed that this transcription factor actually prefers to bind to DNA probes when the DNA probes are methylated compared to unmethylated DNA. So these are in vitro studies using protein binding microarrays and synthetic DNA, but also we can see the same kind of trend in vivo, right? So if we look at, for example, ChIP-seq data from CBP beta and look at the binding sites, we can see that there's a bimodal distribution of methylation in these binding sites. So there are peaks that have very low CpG methylation. There are peaks that have very high CpG methylation. And actually, if we look at these different peaks, these two classes of peaks, we see that different sequences are enriched in them. So this transcription factor seems to bind to this particular sequence, TTGC, with some preference for A at the end, when there is no methylation. But when there is methylation, it prefers to bind to sequences that have a CpG in the middle. And because we know CpG is methylated, so this means that this transcription factor actually prefers methyl CpG binding sites when the DNA is methylated. So now the question is to what extent this is just an exception with a couple of examples such as CBP beta, or this can be more general. So there are more transcription factors out there that can identify and bind to methylated DNA. So one of the earliest larger scale efforts that I know of, and I know that there has been a lot of research on this, and probably I'm not aware of all of them, but one of the earlier ones, I believe, is this study from 2013, where they tried to do mass spectrometry to identify transcription factors that can preferentially bind to methylated DNA probes. So we have methylated and unmethylated DNA, and then they use Celex together with mass spectrometry to see which transcription factors preferentially bind to methylated DNA. And these are the ones that they found. So in mouse embryonic stem cells, there were quite a few transcription factors that seem to preferentially bind to methylated DNA. Obviously, this kind of experiment doesn't tell us how these transcription factors recognize methylated DNA, in what kind of context, what kind of sequence, methylated sequence is recognized by these transcription factors. But this is a good start telling us that there are probably more transcription factors that can recognize methylated DNA. And then there is this other study from 2017 from Karolinska Institute, and I think this is probably one of the landmark studies in this area, where they tried to systematically identify the sequence preferences of transcription factors along with their methylation preferences in vitro using an approach called Celex, which I think many of you might be familiar with. So basically, this is an in vitro evolution experiment where you have a pool of DNA sequences with random sequences, and then you iteratively select the DNA probes that are bound by a specific transcription factor. You enrich the DNA probes that can be bound by that transcription factor, you sequence them, and then you can figure out what kind of sequence is enriched in those probes, right? So what they did in 2017 was that they performed Celex, but on two different DNA library pools, one methylated and one unmethylated. And then they systematically did this for several hundred different human transcription factors, and looked at the motifs or the sequence preferences that would come out of each of these different Celex experiments, methylated or unmethylated Celex. And by comparing these binding profiles, they could tell whether that transcription factor prefers methylated sequence or not. So in this case, when the DNA is not methylated, the transcription factor binds to a CpG containing motif. When the DNA is methylated, it binds to something that lacks the CpG, right? So it tells us that this transcription factor is trying to avoid methylated CpG, so it doesn't like binding to methylation. But then when they applied this method systematically to several hundred human transcription factors, they found examples such as OCT4 that actually preferred methylated CpG sequences based on graphs like this that says that in methylated DNA library, CpG containing sequences are preferentially bound by these transcription factors. And when we look at the overall statistics of all the different transcription factors that this study analyzed, it's very surprising. So about a third of all human transcription factors that they analyzed, they actually preferred methylated DNA over non-methylated DNA. So they call it method plus transcription factors. These are transcription factors that prefer methylated CpG sequences. And then about 23% were methyl minus, so they tried to avoid methylated CpGs. And then about a third of transcription factors did not recognize CpG containing sequences at all. So these were not affected by methylation, obviously, because methylation did not occur in their binding site, right? And then there were other categories like lethal effect and multiple effects. But I think this is the most interesting part, where such a large fraction of all human transcription factors seem to be binding to methylated DNA. And because of studies like this, there is now this emerging view that methylation is not always an inhibitory mark for transcription factors. So there are transcription factors that actually don't bind to unmethylated DNA and preferentially bind to methylated DNA only, or transcription factors that might bind to a particular sequence when the DNA is not methylated, but then their preference changes when the DNA is methylated and they bind to a different sequence. So now the question is, to what extent these observations that are primarily in vitro can be extended and translated to the in vivo behavior of transcription factors? And the idea is to use data such as ChIP-seq experiments, for example, to figure out the preference of transcription factors in terms of binding and recognition of methylated DNA in vivo. But this is not a trivial task. So you know that, for example, when we do a ChIP-seq experiment, we can take the peaks and then we can look at the sequences that are enriched within those peaks, and that gives us the sequence preference of transcription factors, right? So basically, we can look at transcription factor sequence preference just based on the ChIP-seq data alone. But it's not easy to do the same thing for methylation preference because there are a lot of confounding factors. We know that DNA methylation very strongly correlates with DNA accessibility. And we know that DNA accessibility actually affects the binding of transcription factors. So this creates this confounding effect of accessibility, which makes it very difficult to figure out the role of methylation, intramotif methylation, on the binding of transcription factor. We also have the regional methylation. So different CpGs within the same region are often highly correlated. And when we are looking at intramotif methylation for the transcription factor binding site, we have this confounding factor of the methylation of the adjacent regions. So all of these makes it very difficult to basically extract methyl preferences of transcription factors by looking at in vivo data. And this is where my student Aldo comes into the picture. So the work that I'm presenting here, the vast majority of it is actually his master's thesis. So together with Aldo, we came up with this model that we call JAMS, Joint Accessibility Methylation Sequence Analysis, where we try to quantitatively explain the transcription factor occupancy that we see in ChIP-seq data based on all these different factors that might influence the transcription factor binding strength or occupancy. So what are these factors? We have the intramotif methylation that we want to look at. Then we have the sequence of the binding site that we want to consider when we are doing this modeling. The flanking base composition and CpG methylation are factors that can affect the binding of transcription factor. And then we have the DNA accessibility profile around the binding site of the transcription factor that we want to consider. And the idea is that if we include everything into one model that tries to explain the transcription factor occupancy signal, then the model should in principle be able to divide the role among all these different factors and basically deconvolve their effect on transcription factor binding, and at the end give us the effect of intramotif CpG methylation on transcription factor binding, which is what we are interested in. I will not go through the mathematical details of this computational model, but I will just say that basically what we're trying to do is to figure out how accessibility, methylation, and sequence affect the background signal that we get from ChIP-seq, and then the signal that we get from a transcription factor pull down, we model basically as that background signal plus the transcription factor specific signal, which is again itself is modeled as a function of accessibility, methylation of sequence. And by this joint modeling of the control and pull down experiment, signal, and then by considering the accessibility, methylation, and sequence, we can specifically figure out the role of these variables in determining the transcription factor binding strength or occupancy. This model JAMS, we have a computational framework, and we have implemented it in this package called JAMS. So there's a link here in case you are interested to give it a try. We can use this link. But I think the first question is, does it work? Actually, can we identify methylation preferences of transcription factors using this model? And I'm going to start with some examples of transcription factors whose methylation preference is known, basically. So again, CBP beta. I mentioned that this is a methyl plus transcription factor prefers to bind to methylated DNA. And this is exactly the same thing that JAMS also infers from looking at ChIP-seq data from CBP beta. You are familiar with this kind of logo. This is a motif logo. The letters basically tell you what kind of sequence the transcription factor likes to bind when the DNA is not methylated. And then there are these arrows. These arrows tell you how these coefficients or these preferences change if the DNA is methylated. So for example, this upward arrow says that if this cytosine here is methylated, then there is actually an increase in the transcription factor occupancy compared to the baseline level of unmethylated DNA. And we also developed this other kind of visualization. It's a type of dot plot where the circles basically tell you the base preference. So this red dot here, for example, tells you that at position 10, the transcription factor likes to bind to A, and the blue dot says that at this position, this transcription factor doesn't like to bind to G. And then you have these dumbbells here, and these dumbbells are basically the effect of DNA methylation, CpG methylation. So red indicates increased affinity. If this red dumbbell here says that if you have a CpG in this position, its methylation increases the affinity of CBP beta. So CBP is one example. Then we have transcription factor NRF1, which is another example of a known transcription factor with known preference. This one, we know that it does not like to bind to methylated DNA, and actually that is the same thing that we can infer by using JAMS on ChIP-seq data. More examples in the slide. MAX is a known methyl minus protein, doesn't like to bind to methylated DNA. And Kaizo is a well-studied methyl plus protein that likes to bind to the DNA. And you can see that these are the same things that we can infer by JAMS. So the red dumbbell and blue dumbbell, these indicate the binding preference. And the other thing that we asked was whether we can actually predict differential binding of transcription factor across different cell types using these JAMS models. So the idea is that the model includes dynamic features such as DNA accessibility and DNA methylation. And these are the things that change from one cell type to another cell type. So if the model has really learned how these features affect the binding of transcription factor, it should in principle be able to predict the differential binding of transcription factor across different cell lines. The idea is that we train the model on one cell line, on the ChIP-seq data from one cell line, and then we can use that model to predict the signal across different cell lines. And then if we compare these predicted signals, its difference should reflect the actual difference in transcription factor binding. And that's actually what we see. So again, let's start with CBP beta. We can see that there are a lot of binding sites that are differentially occupied by this transcription factor between these two cell lines, HeLa and HepG2. And this observed change in binding is actually very strongly correlated with the change in binding that is predicted by the JAMS model of CBP beta. So this is, in my opinion, a very good prediction, given how difficult the prediction of differential transcription factor binding is. More examples showing that the model works. So we have MAX again. For Kaizo or ZBTB33, we have some of the lowest prediction powers, but still, I think this is quite good. The Pearson correlation is 0.44, which is not that bad, right? So we can predict differential binding, but then the question is to what extent this differential binding and what we predict is actually driven by DNA methylation, because our models contain both DNA accessibility and DNA methylation, and DNA accessibility can be very different between two different cell types. And I wanted to isolate the effect of methylation on this differential binding. So what we did was we only looked at the peaks that had very similar accessibility between these two cell lines. And then only for these peaks, we looked at the correlation between what JAMS predicts and what we see experimentally in terms of differential binding signal. And we see that, again, there is a very good correlation. And because the only thing that is now different between these two cell lines for these particular peaks is methylation, we can basically say that it's most likely that methylation is driving the differential binding of these transcription factors, which can be predicted by JAMS. So all in all, it seems like JAMS is doing a good job at identifying methyl plus transcription factors, identifying their signal. So now what Aldo did, which I think was a very Herculean effort, was to systematically apply JAMS to as many ChIP-seq datasets as he could get his hands on. So 2,200 ChIP-seq datasets from different sources, including ENCODE and several different high throughput papers that looked at ChIP-seq and ChIP-exo data from specific families of transcription factors. So these included about 600 transcription factors. And then we also needed methylation data, which means whole genome bisulfite sequencing, and DNA-seq data for DNA accessibility across different cell lines, so at least 6 cell lines. And then he combined these data to run JAMS for every transcription factor, quality control of the models that were trained in order to figure out which models are actually doing a good job, which models are not doing a good job in terms of predicting the transcription factor occupancy signal from ChIP-seq. And at the end, we came up with 260 models, high confidence models for 260 transcription factors. And if we look at these 260 transcription factors for which we could infer their methylation preference, they come from various transcription factor families. And obviously the most common one is the C2H2 zinc finger family. This is the most common transcription factor family in the human genome, which then I'm showing here broken down into subfamilies, C2H2 zinc finger proteins that have a crab domain. These are very common. About a quarter of all human transcription factors are from this family. So we're seeing a diverse picture from all different transcription factor families in terms of their methyl preference. But then what are these methyl preferences? So if we use the JAMS models to actually classify transcription factors, what we see is that almost half of all transcription factors for which we had a good model are methyl minus transcription factors. Almost a quarter show no effect. And when I say no effect, it means that there is a CpG within the motif, but methylation of the CpG does not seem to make a difference here. And then there are a relatively small number of transcription factors that actually prefer to bind to methylated DNA. So this is a bit in contrast to what we were seeing in vitro, right? And I will get back to this and discuss this a little bit more. So now, first, let's see if these inferences are actually reliable. So what we did was, for those transcription factors that we analyzed and also had Celex data, methyl Celex data, or bisulfite Celex data, we tried to do a comparison. And what we see is that there is actually a fair agreement between these two methods. So let's look at, for example, methyl minus predictions for JAMS. So whenever JAMS says a transcription factor is methyl minus, if we look at the corresponding Celex data, we see that for about 80% of those transcription factors, we also see a methyl minus effect in vitro, right? So this is positive predictive value. The fraction of the things that you predict that are actually consistent with experimental observations or ground truth. Similarly, for methyl plus transcription factors that JAMS identifies, when we look at those few that also have Celex data, we see that for 80% of them, Celex also says that these transcription factors are methyl plus. So again, we seem to have about an 80% positive predictive value here. And when we look at the individual nucleotides within the binding sites that are predicted to have an effect on the transcription factor binding, again, we see a consistency between Celex and what jams infers in vivo. So I won't get into the details because of time. But then what I want to highlight here is that even though we have good positive predictive value, what we see is an overall trend toward reduced methyl CpG binding in vivo. So we were looking at the columns previously, which were the JAMS classifications, and we were seeing how they correspond to Celex classifications. So now let's do it the other way around. Let's look at the rows. So look at the Celex classification and see what happens when we look at how JAMS is classifying, for example, methyl plus transcription factors. So you see that a lot of the transcription factors that show a methyl plus effect in vitro now are showing almost no effect in vivo, or at least we are not able to detect any statistically significant effect based on the in vivo data, right? So there is a shift from methyl plus to no effect. And then let's look at the no effect cases. There seems to be a shift from no effect to methyl minus when we go in vivo, right? Whereas for methyl minus cases, there is no shift. So in vitro methyl minus remains methyl minus, in vitro methyl plus becomes no effect, and in vitro no effect becomes methyl minus, right? So there's an overall shift, and there are different ways of interpreting this shift, but we believe one of the likely hypotheses and scenarios is that this is because of competition with other methyl binding proteins, so proteins that have a methyl binding domain. So for example, methyl plus transcription factors, if they don't bind to the methylated DNA with a super strong affinity, when we have methyl binding domains in vivo, it balances out this methyl preference and kind of we see a no effect case, whereas it's only the strongest methyl plus transcription factors in vitro that also show a methyl plus effect in vivo, which means that they can overcome this competition by MBD proteins. And the other thing that I want to highlight here is that we can also have look at all the novel data that we get from application of JAMS to in vivo data. So there are a lot of transcription factors that we analyzed from ChIP-seq that did not have, for example, Celex data whose methyl preferences were unknown. And one thing that is striking in my opinion is that a lot of the methyl plus transcription factors that we identified in vivo seem to come from this C2H2 zinc finger family, and particularly the crab subfamily of C2H2 zinc finger proteins. There's an enrichment in this family, and this kind of makes sense because these are proteins that were originally evolved to recognize and bind to retro elements and these other repeat elements within the genome and to repress them. And these are elements that are highly methylated, so it makes sense for them to actually be able to recognize and preferentially bind to methylated DNA. So because the time is running out, the last thing that I want to talk about is just some word of caution about how to interpret the results of JAMS. And cases where these results need to be taken with a grain of salt. So let's look at the CTCF JAMS model that we got from analysis of CTCF ChIP-seq data. What we see here is that there is a methyl minus effect. So basically CTCF doesn't like to bind to methylated DNA. And this is actually consistent with literature. Previous in vitro and in vivo studies have shown us that CTCF doesn't like methylated DNA. But what is a little bit difficult to interpret is the fact that JAMS tells us there is an effect both in position 2 and position 12 of CTCF when these CPGs are methylated. So CTCF actually binds to sequences that can have 2 CPGs at position 2 and at position 12, right? And JAMS tells us that both of these positions, their methylation reduces the binding of CTCF. Whereas when we look at in vitro data, we see only an effect in position 2. And this is from a study from Gary Stormo's lab in 2017, where they showed that in vitro, if they expose methylated probes to CTCF, only the methylation of the second dinucleotide in position number 2 seems to affect the CTCF binding. And there are different ways of interpreting this. And I think one thing that we should seriously consider is the fact that these 2 CPGs, their methylation is highly correlated. and it becomes a little bit difficult to deconvolve their effects. So when we have more than one CpG within the binding site of a transcription factor, it might become a little bit difficult based on in vivo data to deconvolve their effects. But the other hypothesis is that there might also be some undetected effect of the methylation at the second position on CTCF binding that went undetected in these in vitro studies. And if there's a chance, maybe at the QA session or later, I can talk about this. But when we look at the in vitro data, there seems to be some signals suggesting that, yes, this might be the case, but I think this is something that needs more studies in the future. So I'd like to wrap up by just summarizing what I discussed today. So basically, I talked about JAMS, which is a computational model we have developed that can identify methyl preferences of transcription factors from in vivo ChIP-seq data. JAMS can do a lot of things, including prediction of differential transcription factor binding. And when we apply it systematically to all the ChIP-seq data that we could gather and download, we saw that we can recover the known methyl preferences of transcription factors, and systematic comparison to in vitro models tells us that a lot of these inferences are correct, but also we can capture novel in vivo transcription factor methyl preferences. So most of what I talked today is actually available in our preprint. I have put the link here. There's this preprint basically discusses JAMS, the model, the method and its results. And as I mentioned, most of this work was done by Aldo, who at the time was a master's student in my lab. Now he's a research assistant. And also an acknowledgement of all the other people from the lab who helped with this study and were involved, particularly Senthil, who helped really with the analysis of whole genome bisulfite sequencing data and funding. So I hope I wasn't too much over time limit. Thank you very much for your attention, and I'll take questions.
Thanks so much for this great talk. So if you have any questions, you can write either in the Q&A box or the chat, or you can raise your hand and I can unmute you so you can ask your question from Hamed. There's one question from Erna Magnusdottir asking, hi, very interesting time. I'm just wondering whether the shift towards less binding of methylated CpGs in vivo could be because of the chromatinized context versus the Celex, which I think is done on free DNA.
Very good question. So as I said, we try to consider chromatin accessibility to the extent that is possible, right? So the chromatin accessibility is within the model, and the idea is that by including it in the model, if there's anything because of chromatin accessibility, it gets absorbed into that component, and we can deconvolve its effect from DNA methylation. It's probably not 100% effective when the data is noisy. So for example, when DNA's accessibility data is noisy, there will be then some residual effect from DNA accessibility that may incorrectly be attributed to CpG methylation. But it's actually one of the main advantages of the JAMS model, that by considering DNA accessibility, we were able to deconvolve its effect. So when we exclude DNA accessibility, we see that everything becomes methyl minus. We've done this experiment basically, and we see that there's no way to identify methyl preferences of transcription factors. So hopefully we've done a good job, but we have to see.
Yes, thanks. Okay, I have also another question myself about, are there like any hydroxymethylation, for example, affinity when it comes to transcription factors, or is it just a methyl-cytosine specific?
There is actually. So I think people have done again mass spectral studies and have shown that some transcription factors can bind to that. But I think the data is scarce right now. So we know that there are transcription factors that can recognize it, but unlike 5-methyl-cytosine, where we have the methyl-Celex data and many other experiments based on protein binding microarrays, etc., we don't have that kind of information. And the other thing is that this is a modification that's not very common in vivo inside the cells. So it might not be easy to extract that information using something like JAMS by looking at the in vivo transcription factor occupants.